Hello students, welcome back. Today in this video we will be going to start with the prokaryotic cell, and in the next one we will be completing the eukaryotic cells. So what basically prokaryotic cells are? As I have already told you that prokaryotic are the simpler one as compared to the eukaryotic cells, and basically they are unicellular in nature. Okay, they are very much simpler, not the complex one. They contain a single circular DNA, and they lack well developed organelles. So in this video we will be completing all these things that include the external uh, structures as well as the cell envelopes. So before starting that, let's discuss what are the characteristics of a living organisms. How we can say that this thing is living or not. So first one is growth. Growth and development should be there. If we compare these things side by side along with the human, for example we take human, so growth and development should be there, reproduction should be there, that means heredity, that means transfers of one's characteristics to the progeny. Okay, it should have metabolism functions, that includes the chemical and the physical processes, that means that includes the anabolism as well as catabolism. Fourth one, movement. Movement is a necessity. It doesn't mean movement that means moving from one place to another, it's also mean the communication. Cell support should be there, like for example in us we have a skeleton system, or we have skin as a protective barrier. So any living organism should have a certain cell support, that can be vacuoles, that can be inclusion body like a storage body. Okay, that can be cell wall. Okay, now the transportation is must. Transport across the cell membrane, so what is needed by the cell can be obtained from the outer environment, and what which is not required for the cell can be releases into the outer environment. Now we will start with the basic chart that we will study in this prokaryotic cells. So it contains the external structures, it contains cell envelope, then it contains internal structures. So this is the chart showing the basic structure of prokaryotic cell. If we talk about the external structure, we have outer appendages. Then we have glycocalyx. Glycocalyx is a covering that is made up of sugar and protein. Then in appendages we have flagella, cilia, pili, fimbriae. And in glycocalyx we have capsule and slime layer. Now if we talk about cell envelope, envelope means a covering. So the one which is covered the whole cell is cell membrane as well as cell wall. And if we talk about the internal structure, it have cytoplasm, it have ribosome for protein synthesis, it have inclusion body that are basically the storage bodies, it have nucleoid instead of nucleus, it have uh, cytoskeleton, that is the basic support that provides support and help aid in transportation, and it have endospore help during the extreme environment. Now I'll be showing you the diagram of prokaryotic cell. So as you can see in the diagram contains flagella that aids in movement. It contains cilia and fimbriae that helps in the attachment. It contains pili, again helps in the transportation of partial DNA transfer. Then we have slime layer as well as capsule. They are both the part of glycocalyx, that is the outer covering which is present outside the cell wall. It contains cell membrane which is covered around the cytoplasm. Then it contains cell wall. The outer membrane is known as cell wall. Then it contains, if we talk about the internal things, it contains mesosome. What is mesosome? It is basically the folding of cell membrane. Then cytoplasm, gel matrix. Then it contains inclusion body that is known as storage body. Ribosome is there. actin cytoskeleton that is providing the basic cell structure or support to the cell. Then it contains nucleoid and also the plasmid. I missed that one in over here. So what basically plasmid is? Plasmid is extracellular chromosome. Now we will start with the external structure. If we talk about external structure, we have appendages that is present outside the cell membrane. So these appendages are for motility, that is flagella, that aids in movement, and for attachment, or we can say communication in between the cells, or they act as a channel, that is pili, cilia and fimbriae. And if we talk about the glycocalyx, glycocalyx basically is a surface coating that is present outer side the cell wall. It can be slime layer or it can be capsule. If the combination is tightly packed, then it is capsule, and if the molecules are loosely packed, then it is slime layer. So firstly, flagella. Flagella is basically made up of three things. It have a basal body, it have hook or we can say curved sheath, it have filament that helps in movement. This is the diagram. So the basal body is a stack of ring that is anchored within the cell wall. They are the stack of C ring that is anchored within the cell wall. Then to basal body we have a curved sheath like structure, a hook like structure that is known as curved sheath. That is a connection or intermediation between the basal body as well as the filament. And now finally the filament. Filament is made up of protein that is known as flagellin. That's why the name flagella came from. It is a long helical tubular structure. So basically it's this filament that helps in the movement of cell in the environment, just like the tadpole move in the water. Now we will study the function of the flagella. So as I have already told you that they help in the movement of cell. So this movement happens in response to a certain stimuli or an external signal or an external stimuli. So it can be chemical based, or we can say chemical stimuli, or it can be a light stimuli. So chemical movement in response to chemical is known as chemotaxis, and the light stimuli, so the movement in response to light is phototaxis. Taxis here terms refer to the movement. Now again if we talk about the chemical or either the light stimuli, the movement can be a positive or negative. So if we talk about chemical, it can be a positive chemotaxis or negative chemotaxis. If we talk about light, it can be a positive phototaxis or negative. Positive means movement in the direction of stimuli. So on the basis of presence of flagella on a cell, the bacterial cells are divided into certain categories. First one is monotrichous. Mono means single. That means a bacterial cell having a single flagella at one end. That is monotrichous, like this. Now the second is lophotrichous. Lopho means a bunch of flagella at single end. So small bunch is there, just like the hose tail. So this is lophotrichous. Third one is amphitrichous. Amphitrichous means flagella at both ends. And lastly, peritrichous. Peri means all over the surface. So peritrichous means the flagella is present all over the bacterial cell surface. So if we talk about the speed, which bacteria move faster. So the lophotrichous move faster, then comes the monotrichous, then comes the amphitrichous, and lastly the peritrichous. Because of the presence of flagella all over the surface, the movement of peritrichous is very slow. Now the fimbriae or cilia. So the basic function, they are the fine proteinaceous structure, or we can say they are the hair-like bristles that's present all over the cell surface, just like the Velcro tape. Their main function is the attachment or the adhesion of cell to the other cell. So it aids in transportation of material, also in the communication in between the cells. So these are the hair-like structure, and just like these two cells are attached towards each other with the help of cilia or fimbriae. Next we have pili. Pili is again a tubular structure present outside. It's a rigid tubular structure made up of the protein known as pillin. That's why the name pili. And this structure is found basically in the gram-negative bacteria. So the cells containing the pili is known as F plus, fertility positive, and the another one is F negative. So it basically help in the partial transfer of DNA. Partial transfer means here, using the pili, one cell transfer its plasmid to the another cell in the process known as conjugation. So the pili connecting the two cells is known as mating bridge. When we study the gene transfer, I will teach you about the conjugation process. Now, glycocalyx. So what glycocalyx is? It is basically the surface coating that is present outside the cell wall. It is made up of protein and sugar molecules. So on the basis of their arrangement, if the protein and sugar molecule are tightly packed, then they are known as capsule. If the protein and sugar are loosely packed, then they are known as slime layer. Their function, it protects the cell from nutrient loss as well as from dehydration. It also prevents bacteria from phagocytosis by the WBC. So it is providing the antigenicity to a bacterial cell, or we can say pathogenicity. Or they are also help in the attachment. That means the slime layer helps in the formation of a biofilm around the cell wall that helps in the attachment of particular cell on certain surfaces. Now the cell envelope. What is cell envelope? Like an envelope carrying the letter. So we can say the content, all the content of the cell is surrounded by a certain envelope. So external to cytoplasm it is present. They can be of two types. Cell wall, that is the outer membrane, then cell membrane, that is the inner membrane. So they both act as a protective layer. It maintains the integrity of a cell. That means it is providing a certain shape. It acts as a protective barrier like our skin, which acts as a protective barrier for our muscles, our internal organs. Now the cell wall. So basically what cell wall did, it determines the cell shape. Like it can be a spherical or cylindrical. It prevents lysis due to the change in the osmotic pressure. That means if the osmotic pressure around in the environment changes, it prevents in the lysis or the collapsing of cell. The primary content of cell wall is peptidoglycan. What is peptidoglycan? It is made up of two things, peptide and glycan. It is a unique framework, or we can say the macromolecular framework of long glycan chains attached or cross-linked with the help of certain peptide fragments. So that basically what peptidoglycan do. It also provides the flexibility to the cell. Now on the basis of cell wall, Christian Gram is a scientist who discovered the gram staining. So on the basis of gram staining, we have divided the bacterial cell in two categories. That is gram-positive bacteria or gram-negative bacteria. When we do a gram staining, it contains a series of dye. It contains series of dye that will give the purple color to gram positive bacteria and pink color to gram negative bacteria. The concept behind this one I will teach you in the further slides. So gram positive is purple colored stain and gram negative stains pink. Gram positive contain thick wall of peptidoglycan, whereas the gram negative contain a thin wall of peptidoglycan. And other than the peptidoglycan, it also have an outer membrane that is made up of lipids, lipopolysaccharides or lipoproteins. Now look at this diagram. Here you can show this brown part. That is the peptidoglycan layer. This is the gram negative one and the gram positive one. The gram positive contain a thick layer, whereas the gram negative contain the thin layer. On the outer side of the thin layer peptidoglycan, we have an outer cell wall that is made up of lipopolysaccharide. Its structure is just like cell membrane, and it contains porin. Porin are like the transport channels that helps in the transport of certain molecules from in and out of the cell. Now, gram positive cell wall. If we study about the gram positive cell wall, what are the key points of that? So, this is the diagram, the thick layer of peptidoglycan. And this peptidoglycan layer is 20 to 80 nanometer thick and is a homogeneous sheet. Why homogeneous? Because it is solely made up of the peptidoglycan molecules. It functions in maintaining the cell integrity and also help in the enlargement during the cell division. We know when cell divide by binary fission, that happens in that case. It also helps in the movement of cation across the cell envelope. And also it stimulates certain specific immune response. If we talk about the pathogenicity, all cells have their different pathogenicity. All bacterial cells have their different pathogenicity. Now the gram negative bacteria. So it contains a thin layer. This the red one is the thin layer of peptidoglycan. The blue one is the outer membrane outside the peptidoglycan layer, which contain porins, and the green one over here is the cell membrane. The outer membrane plus thin peptidoglycan membrane. This outer membrane is made up of lipopolysaccharide as well as the lipoproteins. And also this outer membrane secretes certain endotoxin during the cell infection. It contains lipopolysaccharide. Lipopolysaccharide means lipids plus carbohydrate. Lipoproteins mean lipid plus proteins. And also this help in providing the certain pathogenicity to bacterial cell. And it also contains porins that help in the transportation of molecules from in and out. That means it regulates transport. Now, the gram staining. Gram staining is a microbiological method through which we can identify and classify bacterial cells. It also helps in the classification of the shape, how the cell look. Either it is cylindrical or rod shape or spherical shape. It also helps in the diagnosis of certain disease. That means they are used in the pathological labs for diagnosing infections. So on the basis of this gram staining, we have categorized bacterial into gram positive and gram negative bacteria. Gram positive stain violet and gram-negative stains pink. Now this is the diagram showing the methods through which we can perform the gram staining. So here we use 4 type of dyes, that is crystal violet, then gram iodine, then alcohol, and then saffronin. So crystal violet is the primary stain and saffronin is the counter stain, which actually show the result whether the bacteria is purple or pink. So for the first step, we have taken the slide containing the bacterial cell. Then what we have done, we have poured the crystal violet. After retaining the crystal dye, both the bacteria, either gram positive or negative, stains purple. Then we add gram iodine. Gram iodine is a mordant that will stabilize the dye inside the cell. Then on third step, we are using alcohol. Now as we all know, lipid dissolve in alcohol. So just imagine if the layer is thick, if we have a thick layer of lipid, then it will dissolve slower as compared to the thin layer. And over here we can see that the gram negative have the thin peptidoglycan layer. So what will happen in the presence of alcohol? It will make pores inside the cell wall, and through which what happen, all the crystal violet dye that is inside the gram negative cell wall will came out of the cell through these pores. So at the third step, what we have that gram positive are still containing the crystal violet color or violet color, and whereas the negative is colorless. Now the counter strain. So the cells containing violet color does not retain the pink color, but the cell which is transparent contain the pink color. So that's how we can classify the gram staining, and that's how we can say that. Okay, now the atypical cell wall. Some bacterial cells also have atypical cell walls. That means they may contain no cell wall and they may contain inappropriate or improper cell wall. So some bacterial groups lack a typical cell wall. That are examples are Mycobacterium and Nocardia. And also some bacterial cells do not have cell wall. Example are Mycoplasma. But what they do have, they have their outer surface made up of sterols. That's not the actual cell wall. So their outer membrane is stabilized by sterol. And they are pleomorphic, as they do not have proper skin-like structure or wall-like structure, so they are pleomorphic. That means they can change their shape easily. Now to the inner membrane, which is known as cell membrane. So it is a phosphobilipid layer. We have the phosphobilipid layer that is made up of phospholipids. That is phosphate plus lipid. We have long chains of lipid with the phosphate head. It is also known as fluid mosaic model. Why? Because in this membrane we have proteins embedded internal or external to the membrane, and they are movable. They continuously move inside the cell membrane. They function as it provide the site for energy synthesis, like in case of plant cells, or in the case of bacterial cells also, like in the case of cyanobacteria, they perform the function of energy synthesis. They also help in transportation of the molecule from in and out of the cell. They contains receptor which act as a signal receptor. Whenever in the case of signal transduction some signal is coming from the outside, and they receive that, and further secondary processes happens inside the cell. Now the diagram of phospholipid bilayer. So this is the phospholipid bilayer containing the hydrophilic head and hydrophobic tail. It also have the internal protein and external protein that is embedded inside them. They also have the transmembrane protein. Transmembrane protein also act as a channel, which is a transported channel, and also they act as a receptor. Also they contain some carbohydrate receptors and other receptors also, which is help in the signal transduction that will receive the outer signal towards it. So these molecules, these proteins will randomly move inside within the cell wall. That's why the name given. Thank you. If you like this video, then kindly please subscribe and support. And in the next video, we will continue with the internal structures of bacterial cell. I hope you like it.